All right, and look at this one slide that we looked at last time and then move on from here. So including today, you've got 4 stunning genetics lectures left. I know you're all really already starting to miss it, even though it's not over yet. Yeah, today, then 2 next week, then there's Thanksgiving, and then you got one more. So that's 4 if you count them up. Yeah, the Tuesday after Thanksgiving is the last day of class. So if you look at what we've got planned, you can get overwhelmed in your mind because there's a lot of stuff we're going over. But for the last set of material here, I am carefully picking and choosing, and I apologize, I need to get the something came up again this morning, and so I need to get the exactly what we're going to be reading in each in chapter 15 posted for you. But I'll do that right after this. So there's a lot of stuff, but I'm leaving out many details. So yes, you're going to have to read through the chapter material, but look at the detail that we go over in class and in lecture. That's what I want you to know. I mean, I think you know by this point that I don't pull stuff from the book to ask you questions about, like detail stuff, that we don't talk about here in class. For example, when we talk about translation in chapter 15, there's a whole bunch of information, all of it good, about initiation factors, elongation factors, things like that, but I'm not going to require you to know what each individual initiation factor or elongation factor does, and I will mention that here in class. So when you're reading through, we're learning about the processes, not learning each individual factor like we do when we talk about it in molecular genetics. All right, so here we go. We're talking about alternative splicing. So we said last time, here's an example where you can have 3 exons and 2 introns, and depending on where the splicing occurs, you can have either a messenger RNA that includes all 3 exons if the splice donor and acceptor between exons 1 and 2 are used, and then the splice donor and splice acceptor between exons 2 and 3 are used. But you can pick and choose. Over here you have exon 2 completely cut out because the splice donor here after exon 1 and the splice acceptor after exon 3 are the only ones that are used. So exon 2 is cut out as part of this big piece that contains intron 1 and intron 2. And I can't remember if I talked about it in class last time or to somebody afterwards, but the way that this works, and we don't go into it in this class, is that there are factors, proteins that bind to the introns and exons that help guide the spliceosome machinery to choose which ones are used in different tissues or different developmental time points, things like that. And so that's how you get either this one here on the left or that one there on the right. And so it's pretty intricate how it all works, and that's why we're not going over it here. All right, and then I finished last time by talking about this. So here's the calcitonin gene. You get 2 different splices, and if you get the splice for thyroid cells here, where you get exon 3, or the intron 3 after exon 3 removed, then exon 4 gets produced, and right after exon 4 there's an AAU AAA cleavage site, and so that's it. You get this cut off. Exon 5 and exon 6 never get transcribed. They're still in the DNA, but they don't get transcribed because the messenger RNA is cut right here, and termination of transcription occurs very closely thereafter. With the brain splice, on the other hand, exon 4 is completely cut out. So exon 3 spliced, or intron 3, exon 3 splice donor here is used, but neither of these two splice sites on either side of exon 4 are used. Instead, the splice acceptor right before exon 5 after intron 4 is used, and so because the entire exon 4 and both introns are cut out, this cleavage site is removed and thrown away as part of an intron, and so the messenger RNA never gets cut. This AAU AAA is thrown away here, and then you get exon 5, intron 5 is cut out, exon 6, and then right after exon 6 you get another AAU AAA polyadenylation and cleavage site being used down there, and so that's why you get a longer messenger RNA. All right. So this is sort of like a review of everything we've been talking about. So here's the promoter. Here is the RNA coding part. Not shown are any sequences, if this was a eukaryote, that are far 5' or down here 3' from the gene. So the promoter is required, as we've seen, whether you're prokaryote or eukaryote, for getting transcription started at the correct spot. Then the pre-messenger RNA begins to be transcribed, and you will get capping, and you will get messenger RNA processing. So you will get splicing occurring to remove the introns, and as soon as the AAU-AAA consensus sequence is reached after the last coding exon, the pre-messenger RNA gets cleaved, the 3' end gets a poly A tail, which is going to be longer than this, about 200 bases put on it. And remember, look back to the previous slide, that also exposes a 5' site of the messenger RNA, pre-messenger RNA still being transcribed, that the RAT1 gets on and can go ahead and take care of that. And so all of this takes place in the nucleus. You have to get capping, splicing, and polyadenylation all occurring in order for the mature messenger RNA to leave the nucleus. So there are going to be cap binding proteins, poly A binding proteins, and remember I said those proteins that get on the intron exon boundaries to help the splicing machinery find where to do the splicing. Some of those proteins hang out even after the splicing takes place, and they are also required to get this messenger RNA out of the nucleus. So there's a big time quality control that goes on here to make sure that no messenger RNA gets out of the nucleus if it's not full length to the 5' end, so it's missing a cap, full length to the 3' end if it's missing a poly A tail, or if splicing has been messed up and it doesn't have the right mixture of proteins still bound to the RNA sequence, it's not getting out of the nucleus alive. And it's a good thing. And it's a good thing. We're done already. All right, so you don't have to memorize this. I'm just showing this so that you can see an entire gene. What? So those of you who are in immunology recognize that this is this is a signaling, an inflammatory signaling molecule, interleukin 2, and so this is the gene for it. So only one strand of the DNA is shown. The other strand would be the reverse complement of this, and so right here is the TATA box. That's where, going back to the transcription chapter, that's where the TBP, or TATA binding protein component of TF2D, or transcription factor 2D complex, binds. That will bring in, along with the other general transcription factors, the RNA polymerase, and then right here is the transcription start site. So this A is the first nucleotide that will get transcribed into RNA. And so the RNA sequence, because this is the coding strand, the RNA sequence matches this strand, but the other strand, the bottom strand that's not shown, would be the template strand. So this is the coding strand or non-template strand. And this sequence matches exactly the messenger RNA that would be produced, except that wherever there's a T, remember there's a uracil instead. But look, right here, this is where transcription starts. So this, between transcription start and the ATG start codon that encodes methionine, the first amino acid in the protein, this is the 5' untranslated region, or 5' UTR. Then you get exons. So exon 1, intron 1, exon 2, intron 2, exon 3, intron, etc., until you get down here to the stop codon. In the RNA, it would be UGA. Go Dogs if you're from Georgia, I guess. Only not this weekend. We want Auburn, right? So then this business, after the stop codon, but before the poly A site, this is all the 3' untranslated region. It gets transcribed into messenger RNA, but because it's after the stop codon, it doesn't code for anything. And here, just a few bases after the AA, see, there's a T, but then AAUAAA right there, is the cleavage site. So here is where the messenger RNA would be cut. Somewhere very close after this, maybe down, maybe down in here somewhere where there's all those, where there's that run of T's, would be where the RAT1 catches up and causes transcription to stop. So somewhere really close after this 3' cleavage site is where transcription would actually stop. And so by the time you get down here, I doubt that that DNA ever gets transcribed into RNA. So the poly A tract would be added to this. RAT1 would bind there at the 5' end and chew that up. So as this is being processed, in front of this A at the ATC there at the 5' end, you would have the 7-methyl-guanosine cap added to the 5' end. Then you would have all the introns removed, stringing. So the sequence of the final mature messenger RNA here would be TTAAT, and then you would go AATTACA, etc. down there, and same thing with all the other introns and exons. And you can see, look, there's a GT at the 5' end of intron 1, AG right there. GT, or GU, and AG are fairly common. They're dinucleotides. They're going to appear all over the place in any DNA sequence. That's why it's that consensus sequence around the splice donor and splice acceptor sites that's important for recognizing those binding proteins. Otherwise, you would get mistaken introns and introns that don't really exist cut out everywhere else. So maybe you've noticed it and maybe you haven't, but one other thing I would want to point out about this is, look right here. The introns are huge compared to the whole product. Remember the picture I showed last time where those loops of introns are just giant compared to the whole thing. There's 2,400 bases here. That's more than this whole thing probably. In intron 3, there's that plus 1,900 bases. So intron 1 is relatively small, but intron 2 and intron 3 are each bigger than the entire actual protein coding sequence there. So capping happens right after transcription starts. While transcription is taking place, splicing can occur, but because of the times at which the proteins get to the splice donor and splice acceptor sites, it's not always directly in a row. So you don't always get intron 1 spliced out first, then 2, then 3, etc. But by the time you get down to cleavage of the pre-messenger RNA, it's already going to be capped, and most, if not all, of the introns are already going to be spliced out. So you have that taking place as we go. All right. So once DNA was figured out to be the genetic material, not proteins, and then RNA was discovered and solidified as the messenger. So RNA is what is used for translation or the building of amino acids into proteins. The trick became, how do you go from the 4-letter alphabet of nucleic acids, A, C's, G's, and U's for RNA, to the 20-letter alphabet that are the common amino acids that are used in most proteins. And there were all kinds of crazy theories. The RNA, some of the most amazing are, and people were thinking of these because they had no idea, the RNA will fold up into a 3-dimensional structure that will make little pockets along the way, and each pocket will bind to an amino acid, and then some enzyme will come by and hook the amino acids together once they were in the pockets. That's just one of the far-fetched, but what are you going to do? You have to try to think of some way to do it, ideas for how translation could go. But bring in the good guy of the Watson and Crick pair. I won't disparage Francis Crick. He was both a brilliant theoretician and a brilliant experimentalist. So we've seen him in figuring out the structure of DNA, and now we see him in what became known as his adapter hypothesis. He said there's got to be some kind of an adapter, some kind of a molecule that can interact with nucleic acid and interact with amino acids to adapt between them, to go from the nucleotide sequence of RNA to the amino acid sequence of proteins. And it turned out that he was right. So scientists looking at cells discovered an abundant type of RNA in the cells that were very short and therefore unlikely to code for proteins, and it turns out these are tRNAs or transfer RNAs, and they're going to be the adapter that Crick hypothesized. So it says on the screen, this is the flattened cloverleaf model. So this is what it looks like when it's all flattened out. So you can see here and there and there and there the base pairing. But this isn't what it looks like in three-dimensional structure. It does show, however, the two important parts of the transfer RNA molecule that I'm going to expect you to know. The first one up here is called the acceptor arm. The 3' end of the acceptor arm is where the amino acid is going to hook. So this is the adapter, right? And down here on the bottom, the anticodon arm is going to contain the anticodon loop. The anticodon loop is going to interact with the messenger RNA through regular base pairing. And that's going to bring the correct transfer RNA with the correct anticodon, as we'll see, bringing in an amino acid. So when we get to the next chapter, codons, we've sort of seen, are the 3-letter words that spell out amino acids. Anticodon is just the reverse complement of that. So the 3 bases that will be able to base pair with it. So this is what a transfer RNA looks like when you flatten it out. Here are 2 pictures, both a line drawing showing where all the base pairing is and a space filling model here showing what it looks like in 3 dimensions. The transfer RNA is sort of an L shape. Again, the anticodon loop is down here at the bottom. That's going to interact with the RNA as part of the ribosome. And up here is the acceptor arm where the amino acid is going to stick on. And this is going to come in and bring the amino acids to the ribosomes. We're going to, yes?
what is the argument that like it's possible? So that's why science is based off this, or like what is
RNA was first before proteins. So in the RNA world model, RNA actually existed first and was the repository of information and also carried out the enzymatic functions. Like before there were even membrane enclosed cells, there were RNAs that could self-replicate. I'm looking for the wrong word there. And let me just hold that thought until we get to the codon table. And so it slowly happens over a very long period of time, but when we get to the codon table and talk about redundancy of codons, that shows that there's an evolutionary mark there as well. Because if the codons weren't redundant, then it would be unlikely that all this could have come about. Okay, so starting off chapter 15, where chapter 15 covers both the way that proteins are synthesized by the ribosome, translation, and also talks about how you go from the nucleic acid language to the amino acid language. We're going to start off with another experiment. So George Beadle and Ed Tatum were working with a kind of fungus called Neurospora. You've probably all experienced this when you go into the cabinet and your bread's been in there a little bit too long and it's got that mold on it, because bread mold is exactly, one of the species of bread mold is Neurospora crassa. And so they were doing what I do in my research lab, which is forward genetics. They were trying to figure out what job each gene in this fungus did, or more specifically, they were trying to figure out which genes were required for biosynthesis or making of amino acids in a step-by-step enzymatic pathway. And so they started out by growing Neurospora on what's called complete media. So complete media has all the nutrition you need. So depending on species, you can make some of your nutritional requirements, or you can have to have them all. So for us, a lot of the nutritional requirements and vitamins, for example, a lot of the nutritional things that we need, we have to get from microorganisms or bacteria. We can't, a lot of the amino acids we can't make ourselves, for example. But wild type fungus can also grow on what's called, don't look at the experiment yet, wild type fungus can also grow on what's referred to as minimal media. This just contains sugar for energy and a very basic carbon source. So Neurospora, unlike us, is able to synthesize all 20 of the amino acids. So there's no amino acids at all in this media. So forward genetics involves treating your model organism with a mutagen that's going to change the DNA sequence and break some genes so they no longer work. Like we did in the lab with the sterile mutants that you did in the lab. I treated worms with chemical mutagens and then looked for ones that were sterile. Here, they were mutating the fungi with X-rays that cause breaks in the DNA, and when the breaks get repaired, sometimes you have mistakes happening. So they took many different strains of the fungi, grew them on complete media, and then each one of these they took, so everybody's going to grow on complete media because it has all the amino acids you need to grow, but then they took each of these and put them on minimal media. In most cases, the fungus grew just fine on minimal media, meaning that none of the genes required for mutation, or none of the genes required for production of amino acids were mutated. But occasionally, you'd get one that would not grow on minimal media. But because they had started out after the mutagenesis by growing all of these different colonies that were picked into these tubes on complete media, even though it didn't grow here, you still had this one to go back to. So it can grow there because you give it all the amino acids. With me so far? So it can't make one or more of the amino acids. So then what they did was they took the minimal media and individually supplemented it with, in 20 separate experiments, all 20 of the amino acids. So on the bottom here, it says an amino acid there. That's the only amino acid that is in that tube. So the tube has no amino acids except for that one. And the only tube in which this mutant was able to grow is the tube that had arginine in it. So this particular mutant can't grow if you give it no amino acids. It can't grow if you give it any of the amino acids except for arginine. But it can grow if you give it arginine only but none of the other ones. So all these down here are negative controls, and here's your experimental. But you didn't know which one it was going to be to start with. So this mutation must affect the organism's ability to make the amino acid arginine, because when you give that back they can grow, but if it's missing, it can't grow. Still with me? All right, so these guys did a massive amount of this work and got a big collection of mutants, some that couldn't grow on arginine and some that couldn't grow on a bunch of these other amino acids. But the next experiment that we're going to talk about on the next slide is using these. So they got many different mutants that could not grow on arginine. But as we'll see, they're going to be different genes. Beadle and Tatum came up with the one gene, one enzyme hypothesis. And they explained it by this experiment, saying that only one gene was mutated in this and only one enzyme was hurt. So these organisms can make all the amino acids except for arginine. And for example, if another one had grown here, that organism could make all the amino acids except for proline. So each single mutation had hit only one gene and hurt only one enzyme. Later, this had to be corrected to the one gene, one polypeptide, because a polypeptide is just the name for a complete protein from amino terminus to carboxyl terminus. One gene, one enzyme doesn't work, and you guys already know why, even though you don't think you do, right? So the bacterial RNA polymerase, who can tell me the bacterial RNA polymerase core enzyme? That's not the holoenzyme with sigma factor, the core enzyme that does the polymerizing of RNA from a DNA template. What makes that up? Subunits, but you don't remember what they, right? Two alphas, a beta prime, and an omega, right? So that's one enzyme to do that job. That enzyme requires 5 different polypeptides to go together. Yes, the 2 alphas are the same polypeptide, but that core RNA polymerase requires 5 different polypeptides to bond together with each other and interact in order to form that enzyme. So one gene, one polypeptide. Because if you do a mutagenesis to see to find organisms or find E. coli cells that cannot do transcription, you could have mutations in the alpha, the beta prime, or the omega, and really the sigma as well. But if you're just looking at the polymerase, any of those 4 genes could be done to that, to go to there. If you're talking to microbiologists again, and maybe they don't say this anymore, but like they haven't said a lot of the other things I've mentioned. So auxotroph is a name for a nutritional mutant like we're talking about here. So an auxotroph is a mutant that needs a certain nutritional supplement in order to grow. And I'm just telling you that so in case you ever see that word. All right, so Beadle and Tatum got a whole ton of arginine. You know, they got mutants that did all these, and then other scientists were looking at each individual pathway, right? So Adrian Serb and Norm Horowitz did experiments that followed up on the Beadle and Tatum experiment, and they got a ton of different mutants that were arginine mutant only. So every one of their mutants were just like this one. They can grow on complete medium when you give them all the amino acids. If you're missing all the amino acids, they can't grow. And if you sub in only arginine but none of the other ones, they can grow on that too, meaning that they are a mutant for arginine. But biochemists had been doing experiments looking at what are called biosynthetic pathways. Here's one that's shown here. We've seen biosynthetic pathways before in chapter 5. You guys thought that you were going to be done with that. Remember the duplicate recessive epistasis experiment where the shell was white, or it was white, or it was brown, and it needed both. And it needed both enzymes in order to work. Well, many biosynthetic pathways have a whole bunch of steps. Here we're just showing 3 enzymatic steps. So we're starting out with a precursor compound. Enzyme A, encoded by gene A, converts that into ornithine. Enzyme B converts ornithine into citrulline. Enzyme C converts citrulline into arginine. And so every one of these mutants, when grown on minimal media that had no amino acids in it, is not going to grow. So this is the control. This is the wild type non-mutant control. Each of these, 1, 2, and 3, is a different mutant that they got. Every one of them did like this, where if you supplement with arginine, they could grow. So if you supplement them all with arginine down here, they can grow. But where in the biosynthetic pathway is the problem? By looking at media containing these other 2, they found out that mutant 1, if you gave it ornithine, could grow fine. Ornithine or citrulline. Mutant 2, if you gave it ornithine, it couldn't grow. Citrulline, it could. Mutant 3, you had to give it arginine, it couldn't grow on any of the other ones. And so where the mutants can grow and where they can't shows at what stage of the biosynthetic pathway they're enzymatically blocked. So this guy, if you give it ornithine, it can grow fine. So that must mean enzyme B and enzyme C are okay because you're not giving it citrulline or arginine in that tube. But if you don't give it ornithine, then it can't grow, which means that enzyme A is defective. If you look at mutant 2, ornithine, you can't grow. If you give it citrulline, then it can grow, which means enzyme C is fine. Enzyme B is probably defective. How do you know in mutant 2 that enzyme A is good and it's enzyme B that's defective? You know because there's only, when you do the mutagenesis, these mutants are rare enough that each strain is going to have a mutation in only one gene. So this has to still be good because you're only getting a mutation in one gene. And number 3 here has to get arginine, which means that even if you give it citrulline, the second to last step, it can't grow, which must mean that this enzyme is bad. And here's the word coming back for you, right? So from chapter 5, when I learned epistasis analysis, in addition to the gene interactions that we talked about, like with the Labrador Retriever coat colors and the squash colors and the snail shell colors and all that, the classical definition of epistasis is that a gene is epistatic to all the other genes that occur later in a biosynthetic pathway. If gene A is mutated, let's say that you have to be recessive, right? Then gene A, if big A is good and little a is no good, and you have to be recessive, homozygous recessive to see that mutation, then this would be having recessive epistasis to enzyme B and enzyme C. And if you have a 10-step biosynthetic pathway, it would be epistatic to every single gene that occurs after it, because it doesn't matter if enzyme B and C are no good. If enzyme A is no good, you don't get ornithine, and enzyme B can't take the precursor compound and run with it. And so that would mean that that in mutant 1, you have a mutation in enzyme A, and enzyme A is epistatic to the rest of them. Here in mutant 2, you can grow on citrulline but not ornithine. That means enzyme B is defective. And so this would be epistatic to enzyme C. Why is it not epistatic to enzyme A? Because enzyme A can still convert precursor compound to ornithine. In mutant number 2, you would just, if it wasn't supplemented, you would just get a bunch of ornithine piling up, and it would never get converted on by into arginine. So that enzyme would still work. It's just not able to go all the way and produce arginine. And biochemically, you can measure whether ornithine, citrulline, and arginine are present, so it's not like it's invisible, like with the snail shell example where you get that duplicate recessive epistasis. All right. So I'm going to go into some biochemistry, but I'm trying to be light here. But we have to go through protein structure, and then I'm going to go into how the translation actually works in a conceptual way, right? So here is a giant amino acid. Hopefully by now in your academic career you are appreciating that biology is just full of a bunch of polymers. And this is another hint about, well, how does evolution work? How do you just pop up DNA? Well, you can make nucleotides first. That's simple. And then nucleic acid is just a bunch of nucleotides strung together. Protein is just a bunch of amino acids strung together, and then the R groups on the sides are different. Complex carbohydrates are just a bunch of simple sugars strung together to make a polymer. And so it's easy to put things together. It's easy enough to evolve a train car, and when you make the train car, then you can put them all together to make trains. All right, so the alpha carbon is the center here, and each protein has direction. So just like nucleic acids have a 5' and 3' direction, proteins have an amino terminus and a carboxyl terminus. So the amino terminus in proteins is going to correspond to the 5' direction in messenger RNA. The carboxyl terminus is going to correspond to the 3' direction in messenger RNA. So generally in proteins, the amino terminus is drawn to the left, the carboxyl terminus is drawn to the right when you're drawing a chain. Each alpha carbon has a hydrogen and then an R group, and these are the hydrophilic side groups, the hydrophobic side groups, and then you have some that are, the hydrophobic side groups can either be polar or, actually the hydrophilic side groups can either be polar or they can have an actual plus or minus charge. I'm not going to ask you to memorize any of the amino acid side groups here. That's for other classes, and actually I think you can look them up probably as easy as anything else. Concepts, how it's put together, that's what I want you to take out of here. All right, so when you're putting nucleic acids together with a polymerase, you are making phosphodiester bonds. That's the bond that holds two sugars together, and each sugar has a base attached to it. Here, the bond that holds amino acids together is a peptide bond. So the peptide bond occurs between the carboxyl group of the preceding amino acid and the amino group of the amino acid that's being added. And it is a condensation reaction. You lose the oxygen off of the carboxyl terminus and two hydrogens off the amino terminus to make a water, and then there you go, it's put together. And this bond formation is going to be catalyzed, it turns out, by the 23S ribosomal RNA molecule. So we talked about one gene, one polypeptide. What is a polypeptide? We'll start right here and then go down simpler. Tertiary structure in proteins is the entire length of one protein, from the amino terminus to the carboxyl terminus, and how that folds up. Let's jump back here. So the primary structure in DNA and RNA is a chain of nucleic acids. In DNA, the secondary structure is going to be the double helix, and then you get to more and more complicated structures when that DNA interacts with histones and other proteins to make chromatin. Here, the secondary structure after the primary structure, well, the primary structure is just the amino acid sequence if you were to pull it out in a straight line. But the side groups and also the backbone of the amino acid chain is going to interact to make secondary structure. So this shown here is called an alpha helix. So it's a helix like DNA, only it's only got one chain in it, not two chains linked together like with DNA. Another secondary structure is called a beta sheet. It's flat. Not going to go into too much more of that in this class. Then so the alpha helices, turns, beta sheets go together to form the tertiary structure, which is what one entire protein looks like. It's only one polypeptide chain. So the example molecule here is hemoglobin. So this is the heme. That's not part of the protein. It will bind to it. So the tertiary structure of the heme protein is that purple molecule there. And then you go from tertiary structure, which is the 3D structure of one complete chain, to the quaternary structure, which is when that chain is fully interacting with others. So hemoglobin has 4 different subunits. Just like I said, the RNA polymerase earlier, I used that as an example, has 5 subunits. So the quaternary structure is the entire enzyme complex coming together. And when we talked about RNA polymerase, not only was there the core polymerase that I talked about, but it also exists as the holoenzyme with sigma factor that can interact until the polymerase is brought to the transcription start site. Yes? So the question is, could you say that the tertiary structure is just the interaction of all the secondary structures, and the quaternary structure is the interaction of multiple tertiary structures? Yes, that is exactly right. Now, there's more to it than the alpha helices and the beta sheets. Sometimes there's, like I said, turns. So it'll be like a helix and then like a loop of protein that goes around to make the next helix or something. And sometimes you get interactions between those loop regions and other ones, but that's picky, and your thing is exactly right that you said. All right. So let's talk about the genetic code, which will appear on the front cover of the final exam. So you don't have, well, I mean, you know, so don't freak out. You don't, I mean, you're just going to have to be able to use it and not memorize it. Oh, you're still. No, don't do it. Don't do it. Oh, yeah. We're going to. Yes. Did I show you any amino acid structures? Did I mention any side groups? No. Did I show any of that? No. Are they in the book? Yes. This is what I'm saying. You don't have to know any of the side groups, which of them are polar, because that's biochemistry. Okay. Well, shoot, I'm recording. I was saying I'm open for donations, but I'm recording this, so that's not true. All right. So let's talk about. So I think you're getting the idea that I'm trying to get you the concepts and how it works, right? So, this is actually genetics too. So I'm not talking about how proteins work. I'm talking about how you get from DNA through RNA to proteins. Okay? So, the genetic code. The genetic code is universal. That means with very few exceptions, every organism on Earth is going to use these same codons to code for each of the amino acids. This is straight up evidence of, it evolved once and then everybody uses it because it's just there. Also, the genetic code is degenerate. And I don't mean that it drinks and gambles a lot. It means that there are multiple codons that code for each amino acid. And here's another, why does it work evolutionary side note, right? So scientists, and there's really good material in the book. Don't do it now. Do it after the holidays are over. But the story about this, and you guys are probably not going to take molecular genetics. It's not offered until 2021. Try to keep the snorting to a minimum, China. But the story about how scientists figured out which codon codes for which amino acid is amazing. And it involves Francis Crick again, and some very good experimentation. Francis Crick working together with Sidney Brenner, who later went on to found the C. elegans field, who just died last year, so he can be Saint Sidney now, right? I'll put him up for sainthood. Anyway, so look, here's methionine is the only amino acid that has only one codon. A lot of the amino acids have 2 or 4 codons, but serine is the example, and there's one other one, I can't remember which, maybe it's valine, you're going to have to look on here, but serine has 4 there and 2 down, and arginine's the other one. They both have 6. Arginine has 4 there and 2 down there. So there's 20 amino acids, and there's 4 different nucleic acids. So if you're going to have words that spell out an amino acid, you have to have the ability to spell 20 words, right? Experiments were done to show that the genetic code was non-overlapping. So that means you can't have one nucleic acid involved in more than one word. So given that, if the words were 1 letter long, you could only spell 4 words, right? A, C, T, or G. A, C, U, or G. If you had words that were 2 amino acids long, you can only make 16 different words, right? And so you're stuck again because you can't spell 20 words when you can only make 16. So if you had 3 amino acids, 4 possibilities for the first position, 4 for the second position, 4 for the third position. Now you have 64 possibilities for words that you can spell. It's a lot more than 20, but 3 is the smallest number of letters that gets you over 20 words. But, and here's the evolutionary thing that you were asking about earlier. It's like originally scientists did not know about the degeneracy, that there was redundancy. But think about it. If only 20 of the 64 possibilities actually encoded an amino acid and the others didn't, that would mean that there, so let's say you need 1 stop codon as well. So 64 minus 20 is 44 minus another 1, so there would be 21 amino acids that encoded something or a stop, and 44 that encoded nothing. Well, what happens if you get a mutation? It's very, very likely, it's more likely than not, because there's 44 out of 64 that don't code for anything, that a mutation would change a word that coded for an amino acid into a word that didn't code for anything. And that would completely bust the protein, right? It's better to have it code for a different amino acid, and that still may break the protein, but you've got a fighting chance, right? Also, because of the redundancy, look, in this one, the third codon there could change to anything, and it would still work, right? And furthermore, I don't have time to go into it here, but the most common types of sequence changes for mutations will change from one amino acid to another amino acid that looks the same. For example, from one nonpolar one to another nonpolar amino acid. And so that even if you're changing from one amino acid to another, the probability that that change breaks the protein and makes it not work is still really low. And so having it set up like this makes it so that you can have changes, because you have to be able to accept a certain amount of mutation rate and not die, because otherwise your genome can never change, and if environmental conditions change, there will be nobody in the population who can thrive in the new environmental conditions, and boom, the whole population is gone. It's dead. All right. So because the code is degenerate, that means there are a lot of synonymous codons, just like synonyms are 2 words that mean the same thing. CCA and CCG are synonyms or synonymous codons for proline. Likewise, there are going to be what's called iso-accepting transfer RNAs. So transfer RNAs each have on them an amino acid. Iso means, oh, we're going back to chemistry. Oh. What does iso mean in chemistry? What's isopropanol? It's an alcohol, but what's the iso mean? It's a branched chain, and are they the same or different on both sides? So it's the same, right? So iso means the same. And so iso-accepting tRNAs are different tRNAs that accept the same amino acid. So you're going to get different tRNAs here that all accept valine, for example. In addition, there is something called wobble. So that's pretty much what it looks like too. That, yeah, they're gonna that was a very good demonstration about what the anticodon loop looks like during a wobble situation that I'm going to describe now. So once scientists figured out that Crick was right and that the adapter in Crick's adapter hypothesis was a transfer RNA, then scientists noticed that in almost every cell type, prokaryote and eukaryote, there were never enough different kinds of transfer RNAs. So there were never like 61 transfer RNAs in the cells. There were a lot fewer. So how can that be? And again, it's not written on this slide, but Francis Crick came up with what he termed the wobble hypothesis, which actually turned out to be true again. The wobble hypothesis is that in some cases you can have a transfer RNA interact with a codon in messenger RNA where the 3 bases don't exactly match up. And it was so in this case the codon 5' to 3' left to right is UCC. So the anticodon now, this is going to be 5' over here on the right, and this is going to be 3' GGA, right? A binds with U just like it does with T and DNA. But at the 3' position in the codon and the 5' position in the anticodon, you can have this wobble effect. There's flexibility, there's some flexibility there. That allows cells to make fewer transfer RNAs, and therefore it's easier for them to grow. It turns out that in most cells, I think in all organisms, they have a bias for which codons they want to use. Yes, all of these can code for leucine, but if you look at E. coli or humans or whatever, and you look at all the proteins they make and look at all the times leucine is coded for, 90% of those is going to be 1 or possibly 2 shared of these codons, and the other 2 are almost never used. And therefore it makes sense that you should be able to do the wobbling, because you don't want to have all these transfer RNAs sitting around the cell that are never going to get used. And so for the more rare occasions, you can have these other transfer RNAs that can actually do the wobbling in there. So here's the wobble table. It says which so this is the 5' end of the anticodon. This is the 3' position of the codon. If the anticodon is C, it can only interact with G. If it's A, it can only interact with U. But if the 5' position of the anticodon is G, it can interact with either U or C at the 3' of the codon. Likewise, a U at the 5' of the anticodon, A or G, and we'll get to this. We'll get to that guy in just a second. So, in addition to the genetic code table, also on the front page of the exam is going to be the wobble table. I'm going to put the position in the codon and position in the anticodon. I'm not going to say whether they're first. So you need to remember that it's the 3' position of the codon and the 5' position of the anticodon that interact and wobble. You need to remember that part. But the rest I'm going to show. So remember when I showed this at the beginning of class today? I'm going to come back to it right now. There it is. So all these weird red dots here are rare bases. So the transfer RNA is transcribed from DNA using just the same 4 bases that are always there. But at these red dot positions, enzymes then change one of the bases to something else by making modifications to it. One of the possibilities that happens, but not always, is that the 5' of the anticodon, because it's 5' up here and coming down that way, the 5' of the anticodon can be converted to a non-standard base that is called inosine. That gives that particular transfer RNA great flexibility. So inosine is a modified guanine or G residue. So it can't bind with guanine. But if you have an inosine at the 5' end of the anticodon, it can interact with anything at the 3' base of the codon except for a G. And that gives it a really good flexibility. And so by wobbling, the cell is able to accommodate the fact that there are 61 codons that code for something without having to have 61 transfer RNAs sitting around the cell while translation is actively going on. That is energetically very expensive. All right. Yes? So let's look here. So if there's a G right here, normally you would expect a C down there, right? And if there's a U, then you're not going to get those 3 base pairs that normally occur between a G and a C. So how does it bind in there? The way that, so it's instead of being straight on, it's tilted just a little bit, but the first and second codons are still there. Also the transfer RNA, as we'll see coming up later in this chapter, is held in place by more than just these 3 interactions. There are also interactions that are more complicated that I don't talk about in this class between the backbone of the DNA and RNA that's in the ribosome, so it's enough to hold it together. So a G and a C, if it's a C, man, it holds it in there really nice. If it's a G with a U, it's not perfect, but there's enough, there is a steric hindrance, but the steric hindrance is not enough to keep it from binding correctly. Is that okay? So the steric hindrance is not enough to make it not come in there. And it's because there are a bunch of other interactions going on that we're not talking about here. Does that make sense? So the problem is there's a really good explanation, and I have a great picture for it that I talk about in molecular genetics, but I don't want to, it's sort of like in physics, when you talk about where an electron is in the electron cloud in general chemistry, I just have to say it's somewhere in this little purple bubble, guys, and you just have to believe me. There's a lot of equations that show that, but we can't, so it's, and it's tough, and that's exactly what you're saying. It's tough for you guys to believe that this works, but it does. I mean, it's okay. We believe. Santa's coming. In genetics, if you want a present, you got to deliver it yourself. All right, so just like we talked about for transcription, for translation, protein synthesis, we're going to have 3 different steps. Initiation, that gets everything set up. Elongation, once everything is set up and the ribosome is moving down the messenger RNA, making a protein. And termination, when you get to the end of the coding sequence, find a stop codon, and the messenger RNA has to then, or the protein, polypeptide, and messenger RNA have to separate from each other. As I said before, 5' to 3' direction in messenger RNA corresponds exactly to N-terminus to C-terminus in a protein. As the ribosome moves down the messenger RNA in a 5' to 3' direction, it is adding new amino acids to the carboxyl terminus of the elongating peptide chain. The ribosome is composed of 2 subunits. Each subunit in both prokaryotes and eukaryotes is composed of multiple proteins and 1 or more RNAs. So this is figure 15-16 from the book. This little thing where it's put together is from an earlier edition of the book, but I keep it because I like it and I don't like that they left it out. So the large subunit and the small subunit come together to make the delicious hamburger that is the ribosome. And it all comes together. We'll see how it gets set up. So the large and subunits have numbers on them. We're only learning the bacterial numbers, so 50 and 30, 50S and 30S. The S stands for Svedberg units, and that is a measure of the speed with which they centrifuge through a matrix. So we talked about earlier density gradient centrifugation, where something centrifuges and every molecule will stop when it meets the part of the matrix where it matches the density. Here we're showing it's how fast does something go down through the whole matrix and hit the bottom. Everything eventually will, but larger items are going to centrifuge through there faster than small ones. So the large subunit in bacteria has 23S and 5S ribosomal RNAs. The small has 16S. And you need to know these because we're going to talk about both what the 16S ribosomal subunit does, and I've already mentioned what the 23S ribosomal RNA does in the large subunit, and that is makes the peptide bond form. The large subunit is referred to as containing what's called the peptidyl transferase center. So up here in the large subunit, when it's together, that's where the peptide bonds are going to be formed, and the 23S ribosomal RNA is going to do it. The small subunit down here at the bottom is referred to as the decoding center, because in pockets within the small ribosomal subunit is where the anticodon loops of the transfer RNA are going to meet the codons of the messenger RNA. So what amino acid comes in here is determined by how the tRNA interacts with the messenger RNA down there, and then the forming of the peptide bonds, adding a new amino acid to the growing protein chain is done up at the top there. So we'll talk about this and then be done for today. The ribosome, as it comes together, is going to have 3 interaction sites for transfer RNAs. So I always draw them in this way, so it's either APE if you're going right to left, or EPA from left to right. But I always draw them in this way because we're assuming the ribosome is moving from left to right across your screen down a messenger RNA, and in that case the A site is going to be interacting first. So as we'll see next time, the A site is called the amino acylated tRNA site. What is an amino acylated tRNA site, or tRNA? It is just a transfer RNA that has an amino acid stuck on it. So the A site, we'll see next time, excuse me, is going to be where transfer RNAs bind when they're bringing in amino acids to be added to the protein chain. The P site is the peptidyl tRNA site. The tRNA in the P site is going to actually have the entire developing protein chain hooked to it. And then the E site is where we'll find out transfer RNA ends up after it gives up the amino acid that's hooked onto it, and that's called the exit site because, not surprisingly, when a transfer RNA without an amino acid hits the E site, it loses affinity for the ribosome and leaves. Okay, so I will get the rest of the it's more complicated on this chapter. I'm going to get the reading assignment for the 5th edition is fine. I just needed to decode the pages for the 6th edition because the page numbers have changed. Please read through the rest of it, but have the slides out while you're doing it so you can see the level of detail that I'm expecting you to know. When we talk about initiation and elongation, you are not going to be required to know what individual initiation factors do and individual elongation factors do and individual release factors do. You'll just need to know what they do as a whole.